Change.
All the
Good evening, students. Last week we discussed about the autoimmune system as well as the lymphatic system. So now this week we will start the immunity part, remaining part of the immunity part. Today onwards I will start to about the AIDS cancer as well as some alkaloids which are extracted from the different plants, which are used as one of the drugs, and the drug addiction and the effects of drugs. Apart from that, alcoholism and the smoking, the effect of alcoholism or alcohol on our body, and the effect of the smoking on our body. So this part is remaining. So today I will start the next part of the immunity, that is immunological disorders. What these immunological disorders are? What do you mean by this immunological disorder? Let us see. Definition. Any situation that results in the impairment of immune response is called immunological disorders. Immunological disorders. That means any situation that results in the impairment of the immune response. Any situations, whatever it may be, the situation might be whatever it may be. So even though during normal situations also the individual is lost its immune response. Otherwise, when the individual is exposed to some harmful chemicals or maybe the pathogens or it might be the antigens, in such condition the immune response goes on decreases. Otherwise, due to defective directions by our autoimmune response, by our autoimmune system, it is going to be misguide our immune response, immune system of an individual, so that may lead to the immunological disorders. So it might be occurs in any situation. So any situation that results in the impairment occurs within the immune response. Impairment occurs in immune response is called as the immunological disorders. So such immunological disorders might be of different types. The first one is immunodeficiency disorder. This is one of the immunological disorder. The second one is hypersensitivity disorder. The third one is autoimmune disorder. The fourth one is graft rejection disorder. So these are certain types of immunological disorders. The first one is the immunodeficiency disorder, where the immune system of an individual is somewhat reduced or it is totally absent. One of the newly born N1 is suffering from the immunodeficiency disorders. Otherwise, the hypersensitivity of disorder. Some of the individuals, they are highly sensitive to the certain allergens. Say for an example, the cigar smoke, which may bring some allergic reactions within the body in an individual. Cigar smoke content, or maybe the tar content, so smoke tar content, which is released by the one of the a person, that will be inhaled by the another person, that may lead to the allergic inflammation in an individual. It brings some allergic reactions within the body, allergic inflammation within the body. So that later on, that slowly it gets it reduces the immune response of an individual against the smoke tar. That may lead to the disorder. That is called as immunodeficiency disorder. Otherwise, the immunodeficiency disorder is mainly because of the various factors, like the gene factor, so mutant gene, or the defect occurs within the gene, the faulty gene, which may bring some one of the immunodeficiency disorder in an individual. Otherwise, the immunodeficiency is mainly because of acquired ones, or maybe the infections, due to the infections, or it might be occurs due to aging. That may lead to immunodeficiency disorder. Apart from that, the hypersensitivity, as I said, hypersensitivity to nothing but the sensitive. The immune response of an individual is highly sensitive to the particular allergens, or maybe particular pathogens. Then autoimmune disorder. I think in the last class, Madam explained about the autoimmune disease, autoimmune disorder. Our own tissues, our own organs are going to be destroyed by our immune response. So that is autoimmune disorder. So for my best example for it is the myotonic dystrophy is one of the best example for this autoimmune disorder. Even though the insulin also. Then the graft rejection disorder. The graft rejection disorder is one of the immunological disorder where after the transplanting the particular organ from the donor to the recipient, after transplanting the particular organ from the donor to the recipient, so that transplanted organ is going to be rejected by the immune system of an individual. That may lead to graft rejection disorder. So these are some the immunological disorders we are going to observe in human body. But today we will start the immunodeficiency disorders. Immunodeficiency disorder. What do you mean by this immunodeficiency disorder? The immunodeficiency disorder is mainly caused by due to the decrease in immune response. Immunodeficiency disorder is a disorder caused due to reduction in immune system or due to reduced immune system or absence of immune system. Immune system might be absent or it is completely reduced. So that is called as an immunodeficiency disorder. So this immunodeficiency disorder might be of two types. One is a primary immunodeficiency disorder. The second one is secondary immunodeficiency disorder. What do you mean by this primary immunodeficiency disorder? The primary immunodeficiency disorder is caused by change in gene. It is caused by the change in gene. That is faulty gene or because of mutant gene. Change in the gene content, change in the gene frequency or due to the faulty gene. Due to the faulty gene. So this is called as congenital disorder. Congenital disorder. Because the change in the gene frequency or maybe the changes in the gene content is occurs at the time of birth only. Otherwise, the mutant gene or maybe the faulty gene is present at the time of birth only in embryonic condition. That may lead to some disorders. So such type of disorders are referred as the primary immunodeficiency disorders. So the best example for this primary immunodeficiency disorder is the severe combined immunodeficiency disorder. That is SCID, severe combined immunodeficiency disorder. It's the best example for the primary immunodeficiency disorder. The second one is the secondary immunodeficiency disorder. The secondary immunodeficiency disorder occurs or is caused due to the infections. Is caused due to the infections or it is due to the aging. Or it is due to the aging or etc. So which is acquired ones. Which is acquired ones at the time of during the lifespan of an individual or any time in an individual, the lifetime of an individual might be it gets affected where the immune deficiency we are going to observe. So this is mainly because of the infections or due to the aging. Due to the aging factor, so the individual's immune response goes on decreases, it goes on reduce, or it is totally in some cases of reaching certain age period, might be after 60 years of age or maybe the 55 years of age, the individual immune system goes on drastically reduced. That may leads to the various diseases. That may leads to the disorders. That is, this is because of the decrease in the immune response of an individual due to the deficiency of the immune response of an individual. So that's why individual might be suffering from the various disorders. They are going to be exhibiting some amount of the many symptoms, the group of symptoms are exhibited by an individual due to the reduction in the immune response. The group of symptoms are exhibited by an individual due to the reduction in the immune response. So that's why, so this is occurs due to the infections or due to the aging. So that's why it is also called as acquired ones. It's also called as acquired ones, not the congenital. The primary ones it is congenital. The secondary ones are not congenital. The secondary ones are acquired ones. Secondary ones are acquired ones. Now, so with the example, if you observe the example of the secondary immunodeficiency disorders, HIV or AIDS, is the best example for the secondary immunodeficiency disorder. Now, let's start AIDS. So AIDS is an example for which type of disorder? It is a secondary immunodeficiency disorder. So they will ask an exam, need exam. AIDS is the best example of. Otherwise, they will also ask the following are the examples for the secondary immunodeficiency disorders. So amongst the option, this will be the one of the option. So we'll go for this AIDS. AIDS is a secondary immunodeficiency disorder, not SCID, severe acute, severe congenital immunodeficiency is not an example for the secondary ones. It is an example for primary ones. But here is AIDS is an example for secondary immunodeficiency disorder. So let us start. AIDS. The abbreviation is AIDS abbreviation is acquired immunodeficiency syndrome. This is the abbreviation of AIDS. This is the abbreviation of AIDS. So this AIDS is one of the secondary immunodeficiency disorder which destroys the entire immune system of an individual. It's called AIDS. It's called AIDS. So here, the AIDS was reported first time in 1981 by CDC USA. By CDC USA. Center of Disease Control. Center of Disease Control in USA. It was first reported by the Center of Disease Control USA in 1981. In 1981. So it is mainly caused by one of the causative organism called HIV. HIV is human immunodeficiency virus. So this HIV is one of the retrovirus. It is one of the retrovirus. Since from last 30 years, it has spread all over the world. It has spread all over the world. Killing more than 25 million people. Killing more than 25 million people. The suspect of AIDS is mainly caused by the HIV virus, the human immunodeficiency virus, or the retrovirus. It is mainly caused from one person to another person, or it gets infected from one person to another person through the various mode of transmission. Through the various mode of transmission. So let us see which are those mode of transmission of this HIV. Mode of transmission. How it is just transmits from one person to another person. How it gets infected from one person to another person. The first one is mainly transferred from one person to another person through the sexual intercourse. Through the sexual intercourse. So whenever the individual are involved in the sexual coitus with more than two female individuals, or it is multiple partners. When an individual, when a single male partner is involving in sexual coitus with the multiple partners, in such condition, due to the exchange of the body fluid, suppose the person is one of the male individual, if it is involving in multiple sex with more than two females, unprotected sex, that too, if he is involving in unprotected sex with multiple partners, in such condition, might be the infected person is going to be transmits the fluid, or maybe exchange the fluid between the individuals, or to the other individual, or maybe to the healthy partner. In such condition, the female individuals, or maybe the healthy individuals, they are going to be suffering from this case. That is, the transmission of HIV occurs only through the exchange of the fluids, not through the physical touch, physical in contact. Not mere with the touch, not with the physical contact. With the physical contact, there is no transmission of the HIV. from infected person to the healthy person. So that's why here it is transmitted whenever the exchange of the body fluid is occurs between the two different individuals. It is occurs between the infected person to the healthy person, with the healthy person. So during the sexual intercourse, so when they are involving in the multiple sex, so with multiple partners, unprotected sex with the multiple partners, unprotected sex with the multiple partners. In such condition, there is a exchange of the body fluid. There is an exchange of body fluid. So when there is an exchange of body fluid through the contaminated fluid, it will be enters into the healthy person body. So this is the one of the chance where the healthy person can suffer from this AIDS. The second one is through the blood. During blood transfusion. So here there is a chances of the transmission of this HIV from the infected person to the healthy person. Suppose one of the donor individual is infected with this HIV. Suppose he is suffering from the AIDS. He is an unknown person. Might be is an unknown. But in such condition, when this individual is ready to donate blood to the another healthy individual, to the recipient, in such condition, the HIV or maybe the viruses which are present in the blood, that is the contaminated blood, when it is transferred to the healthy person, it is donated to the recipient. The recipient might be possessing such type of contaminated blood from the donor, and the individual is suffering from the AIDS also. Otherwise, the blood content and components which are contaminated with this HIV, blood components which are contaminated with HIV. In such condition, during the transfusion of such type of blood, it might be transmitted to the healthy person. So that will lead to the AIDS. The third one is contaminated needles or the syringes. So, suppose one of the syringe or maybe the needle which is used to inject a particular medicine to one of the AIDS patient, if the same type of if the doctor or maybe the experts, if they use the same syringe or needle in order to inject one of the injection of a medicine to the another healthy individual, in such condition, without a proper sterilization method, if they are using the same syringe or maybe the needle to the healthy person, might be it is contaminated with the HIV virus, and that will be gets transmitted to the healthy person. So this is another way of the mode of transmission of this HIV or maybe AIDS. Next one is the fourth one. Mother to children or is developing fetus. If suppose the mother is suffering from the AIDS, but she is asymptomatic one, might be she is asymptomatic, In such condition, her blood plasma might be possessing these HIV viruses. So these viruses are gets transmitted from the mother to the developing fetus. Mother to developing fetus. Mother to developing fetus. Within the womb, it will be transmitted from the mother to the fetus through the placenta. Through the placenta. As you know that the placenta is one of the organic connection between the mother and the developing fetus. Within the womb. Within the womb, the placenta is act as one of the organic connection between the mother and the developing fetus or maybe the developing young one. So through the placenta, the food gets transmitted to the developing fetus. Along with that of various antibodies as well as the various protein molecules. In such condition also, along with this fluid content, even though the HIV viruses which is going to be transferred from the mother to the fetus through the placenta. So that's why the developing fetus or maybe the developing young one is also suffering from the HIV. So these are some modes of transmission of the AIDS or maybe the HIV virus. HIV is one of the transmissible lethal disease. AIDS is one of the transmissible lethal disease. The transmissible, it may be transmits from one person to another person, and that too it is called lethal disease. Why it's called as lethal disease? The individual, those who are suffering from this AIDS, they will definitely die, but we can prevent the symptoms, or we can prolong the death. That's all. We can prevent the expression of the progression of this HIV virus, the replication of this HIV virus, but we cannot prevent the death of an individual. So that's why it's called as a lethal disease. Transmissible lethal disease. The suspect of AIDS is mainly caused by the HIV. It's mainly caused by the HIV. So if you observe the HIV virus, structure of HIV, if you observe, HIV virus is it is a retrovirus, the group of retrovirus, rabies, enveloped protein layer which enclose which enclose two single-stranded RNA genetic material, which enclose HIV is one of the retrovirus enveloped by a protein layer which enclosed two single-stranded RNA genetic material, single-stranded RNA molecules, single-stranded RNA molecule, which is a RNA genome, which act as genetic material. RNA is act as genetic material in virus, where it possessing how many of RNA molecules? It possessing two RNA molecules. It possessing two RNA molecules, which are single-stranded ones, not double-stranded, single-stranded. The single-stranded two RNA molecules are enclosed within the protein envelope. Within the protein envelope. If you observe the structure of this virus, HIV virus, so as I said, it is one of the retrovirus. It is one of the retrovirus possessing the single-stranded RNA. How many single-stranded RNA molecules are there? Two molecules of the single-stranded RNA are there. It is single-stranded. So this question was asked in the exam, NEET exam. In HIV virus, how many the molecules of RNA are there? So might be they will ask the two molecules of single-stranded RNA, the two molecules of double-stranded RNA, or a single molecule of double-stranded RNA, or a single molecule of single-stranded RNA. So here it is single-stranded, the two molecules of single-stranded RNA, two molecules of single-stranded RNA. Let us see. Here it is one RNA molecule, here is another RNA molecule. It is attached with an enzyme, that is reverse transcriptase enzyme. It is in turn covered by the protein layer. This protein layer is in turn covered by another protein layer. Then this viral protein envelope is intercovered by. It is intercovered by the lipid layer. So this lipid layer is somewhat this thick, and this lipid layer is mainly possessing the glycoproteins. It is mainly possessing the glycoproteins. So these glycoproteins are mainly possessing the head as well as the stalk. So glycoproteins are different types, two types. So this one is a glycoprotein. It is GP120 glycoprotein, and here it is the stalk. This one is GP41 glycoprotein. Here this one is lipid layer. Here is protein envelope. And here is genetic material RNA. There is a single stranded RNA, and This one is reverse transcriptase enzyme. This one is reverse transcriptase enzyme. So here in this HIV, how many RNA molecules are there? RNA molecules are 2. The reverse transcriptases are 2, possessing glycoproteins. How many types of glycoproteins? 2 in number. Which are those? One is GP120 and the second one is GP41. So this is the structure of HIV virus, where it is mainly possessing the GP120 as well as GP41 glycoproteins. These glycoproteins, they mainly attack CD4 receptors. CD4 receptors present on TH cells, that is helper T lymphocyte cells. Helper T lymphocyte cells. TH lymphocytes are the TH cells. Otherwise, these are also present on dendritic cells. These are also present on macrophages. Even though, so these are mainly attack on the various other parts of the body cells where these body cells are possessing the CD4 markers. They are possessing CD4 marker. These macrophages, they are possessing CD4 marker. The alpha T lymphocyte cells are also possessing CD4 marker. The dendritic cells are also possessing CD4 markers. So where the CD4 markers of the receptor cells, they mainly attach or binds with this GP120 molecule of the HIV. So once it is gets attacked or attached to the one of the macrophage, or it might be the alpha T lymphocyte cells, then it gets inoculated into the host cell, into the macrophage, or into the alpha T lymphocyte cells, or into the dendritic cells, and thereby they are undergo the rapid multiplication process by which the new daughter viral RNA are going to be produced. Then how this HIV virus, once it is entered into the host cell, or maybe enter or attack to the helper T lymphocyte cells, how it is undergo the replication process. So let us see the mechanism of HIV replication. So this is the entire structure of the HIV virus. So this is the protein coated layer. Here it is encloses the RNA molecule. The two molecules of RNA are there, which are single-stranded ones, possessing to the enzymes, that is the reverse transcriptase enzymes. They are attached to each RNA molecule. And around this protein envelope, there is a presence of the lipid layer. The thick lipid layer mainly possessing the various glycoproteins. So these are glycoproteins. The GP120 as well as the GP41, these are referred as glycoproteins. These are glycoproteins. So glycoproteins are represented with GP. So this is the structure of HIV. Next. Mechanism of HIV replication. The replication of HIV virus. Once the HIV is getting into the host cell, HIV is getting into the host cell. So this HIV virus is mainly releases its genetic material RNA, which is a single-stranded ones. The two molecules of RNA are there. Along with the reverse transcriptase enzyme, it usually attack helper T lymphocytes. Usually attack helper T lymphocytes and also attack macrophages, dendritic cells, even though some other cells of the body also, which are possessing the CD4 marker. So as you know that the helper T lymphocyte cells, so helper T lymphocyte cells are the main lymphocyte cells which has the memory, which is able to identify the invading pathogen, which is able to recognize the invading pathogen. The antigen molecule of the invading pathogen is going to be recognized by the helper T lymphocyte cell because it has the memory and it is in turn triggers some other cells of the body. It in turn triggers the killer T lymphocyte cells present within the blood, otherwise which in turn triggers memory cells, and thereby these memory cells all gets in turn stimulates the B lymphocytes, and these B lymphocytes they directly produces the antibodies against the antigen molecule, by which antigen-antibody reaction is occurs. So because of antigen-antibody reaction, the invading pathogen or maybe the antigen molecule is going to be destroyed by the immune response of an individual, immune system of an individual. So thus we can protect our body with the help of these helper T lymphocyte cells. But these HIV viruses, these are primarily, they are mainly attacked with these helper T lymphocyte cells because it's play a vital role in the cell-mediated immunity. It's play a vital role in cell-mediated immunity. Killer T lymphocyte cells are also involved in cell-mediated immunity. They provide the cell-mediated immunity to our body. And these helper T lymphocyte cells, they are mainly possessing the CD4 marker on their surface. That is, CD4 receptors are present on the surface of these helper T lymphocyte cells. That's why they are easily attracted by the HIV viruses, and these HIV viruses are gets attacked on these helper T lymphocyte cells, and they gets inoculated into the helper T lymphocytes, and they start to undergo the replication process, by which it destroys almost all the helper T lymphocyte cells present in the blood plasma or within the body. So as a result of that, immune response of an individual is somewhat decreases drastically. So when the helper T lymphocyte cells are start to destroy, the individual is going to be expresses primary symptoms of the AIDS. Likewise, it also attacks the macrophages because the macrophages are play a vital role in phagocytic action. They are involving in phagocytosis process by engulfing the foreign body, by engulfing the microorganism, by CD4 receptors. Likewise, so the dendritic cells are also possessing the CD4 markers, dendritic cells which are present in the brain. So that's why, so after attacking all these cells present within the blood plasma or in the tissues, even though these HIV viruses, they are mainly attack some other cells of the body also. Other cells of the body. But these cells should possessing CD4 marker. If they are possessing CD4 marker, then it will be attacked to these other cells. Now, so this is a virus. Here is the RNA genome present within the virus, along with the reverse transcriptase enzyme. So this virus is entered into the host cell. This will be entered into the host cell. Once it is getting into the body cells, now this virus of the entry into the macrophages, into the helper T lymphocyte cell. Assume that this is the TH lymphocyte, TH cell, helper T lymphocyte cell. Okay. Now it releases its genetic material. It releases its genetic material. Along with the reverse transcriptase enzyme. Now this released single-stranded RNA molecule with the help of reverse transcriptase enzyme, it is converted into complementary DNA strand. It is the complementary DNA strand. Now this is the primary complementary DNA strand produced by this viral RNA with the help of reverse transcriptase enzyme. Reverse transcriptase enzyme. Now this viral DNA, now it is a DNA. This is the DNA strand. This is the DNA strand, but it is single strand. It is a single strand. It is a DNA strand. Complementary DNA. It has a complementary base pair against this against the viral RNA. To the viral RNA, the DNA strand complementary is going to be produced as with the help of reverse transcriptase enzyme. Now this DNA strand by using the reverse transcriptase enzyme, the same enzyme, the reverse transcriptase enzyme, it catalyzes the DNA molecule to produce another DNA complementary molecule. Another DNA complementary molecule. So here from this viral RNA, the two DNA strand complementary are produced, but this is a secondary DNA strand complementary molecule produced by the primary DNA strand in the presence of the reverse transcriptase enzyme. Here also the reverse transcriptase enzyme will catalyze here. So with the help of reverse transcriptase catalytic activity, this primary DNA strand complementary is in turn produces the secondary DNA complementary strand. So now these two DNA strands which are complementary, they are in turn modified into the viral DNA. This viral DNA is double stranded ones, not single stranded. This is double stranded. Now, this is the viral DNA. The viral DNA is synthesized by the viral RNA with the help of the reverse transcriptase enzyme. With the help of the reverse transcriptase enzyme. Now, this viral DNA, which is double stranded ones, is gets incorporated with the host cell DNA molecule. This is host cell DNA. Host cell DNA. This is the host cell. This is the helper T lymphocyte cell. This helper T lymphocyte cell DNA is here it is, which is double stranded ones, which is double stranded. Host cell DNA is there. So here this. So this is the host cell DNA. Now this host cell DNA molecule, the viral DNA gets incorporated here. Like this, viral DNA gets incorporated. Now this host cell DNA with viral DNA is now it is undergo the transcription process. Here is reverse transcription process. Here it is the reverse transcription process. Because of the reverse transcription process, viral RNA is converted into viral DNA. Now this viral DNA gets incorporated with the host cell DNA molecule. Now the viral DNA is undergo the transcription process. The transcription process is nothing but the formation of the RNA molecule from the DNA molecule. That is called as a transcription. So here, because of this, what happens here? The many viral DNA RNA are going to be produced. Many viral RNA are going to be produced. Along with this, the reverse transcriptase enzyme. And at the same time, within the host cell is going to be produces many protein particles. These protein particles are gets compiled along with this the viral RNA, which in turn modified into the daughter viruses. So these daughter viruses, or maybe the newly formed daughter viruses, by rupturing the host body, these are comes out from the host cell. The daughter viruses are comes out from the host cell, and these are in turn attack other healthy cells of the body, by which they are undergo the rapid multiplication process. As a result of that, the more and more the helper T lymphocyte cells as well as the macrophages and the phagocytic cells are gets destroyed, results in the rapid deficiency of the immune response of an individual. So thus it will be attacked to the other parts of the body tissues also, where the other parts of the body tissues are will be the cells which are marked by the CD4 receptors. So thus rapidly the entire immune system of an individual is completely destroyed, and thereby individually start to express the symptoms of AIDS. So this is about the mechanism of HIV replication. So in next class tomorrow I will continue the further part.